resource persons. So sir role is also greatly acknowledged for this short-term course. Now I request Dr. Shashikala Madam to introduce our speaker, Dr. Narasimhan sir.
Thank you, sir, Vijay Bhaskar Reddy, sir. It's my great pleasure and honor to introduce today's eminent speaker, Dr. D. Narasimhan Garu. Good morning, sir. He graduated and obtained PhD from Madras Christian College, Tambaram, Chennai. His specialized areas are biodiversity, taxonomy, ethnobotany, medicinal plants, and plant anatomy. He taught and PG for near about 25 years. He published 60 papers in referred journals. He was titled Plant Diversity, Human Welfare and Conservation. He was a co-author to 2 books entitled Commentaries of Chennai, a photo identification guidebook published by Urban Forestry Division, Tamil Nadu and Forest Department, Chennai, and ethnobotany, the Konda, Poraja, Gadaba, and Bonda of the Korapur region of Odisha, India, published by DK Print World Limited, New Delhi. He carried out 10 projects as principal investigator and co-investigator, funded by various agencies such as UGC, Department of Biotechnology, Tamil Nadu Forest Department, Foundation for Revitalization of Local Health Tradition, Ministry of Environment and Forest, and Shastri Indo-Canadian Institute. Developed database on Angiosperms of Tamil Nadu and invasive alien species of Tamil Nadu for the Department of Environment, Government of India, hosted on the website of ENVIS. Documented plant resource Chengalpattu, Javadu Hills, Kolli Hills, Kanyakumari Wildlife Sanctuary, Andaman and Nicobar, documented ethnic knowledge of tribals, Malayalis, tribal community of Koraput, and tribal communities of Andaman. Studied medicinal plant markets of Chennai and Virudhunagar. Guided 19 MPhil and 14 PhD scholars in taxonomy, conservation botany, medicinal plants, and ethnobotany. 2 of the theses has been adjudged as a best thesis in botany, submitted in University of the Madras, and was awarded Dr. R. Kalyansundaram Memorial Prize. Resource person in fields of taxonomy, medicinal plants, ethnobotany, and IPR and environment, and delivered lectures in university colleges and for the voluntary organizations. Honors, he honored with different awards. Out of them, awarded Dr. V.V. Sivarajan Gold Medal given for taxonomist by Indian Association for Angiosperms taxonomy for the year 2008 to 2009. So this is a very brief introduction. If we go elaborative introduction of the resource person, our time will not be permitted, so I have cut shorted his bio sketch, and I have introduced Dr. Narsimhan Garu. Sir, thank you, sir. Thank you for being here, sir, to enlighten our participants. Now the session is yours, sir. Please continue, sir. Thank you.
Right. Sir, kindly. Okay, sir. Yes, sir. So, good morning, everyone. And let me first of all acknowledge the invitation by Dr. Vijay Reddy, who approached me maybe a week ago. And it's my pleasure to join, of course, one of the well-known colleges, one of the oldest colleges in Andhra. And also I acknowledge Dr. Sashikala, who introduced me very elaborately. And good morning, all the other participants. In fact, Dr. Reddy wanted me to talk about this area. Of course, he just broadly left it to me. I said, I'll choose this area. So I thought probably we will go through the history of classification, how different people have classified, what are the parameters they have classified, and how do we approach? And where are we now? Where do we stand now? So this is the kind of thing which I'll be passing through some of the slides. Okay. Right. Okay. Right. In fact, the poster that Dr. Reddy has sent has included these three types of classification: artificial, natural, and phylogenetic, which are age-old kind of terms. In fact, these terms have been there in the history of taxonomy for nearly, I don't know, maybe a century or more. But are these terms still valid? That's one question I raised before I go into the subject. In fact, every classification is artificial. There is no classification that is natural. Because we classify, when we classify, it is automatically artificial. But however, some kind of concepts have crept into our minds, because in those days, what has been considered artificial is basically Linnaeus classification, which was primarily based on only sexual characters. And other classifications which are based on more number of characters, such as Bentham and Hooker or Ray and others, have been considered natural. And later, of course, when Darwin theory emerged, the classification which came afterwards, we started considering as phylogenetic. But any classification, whether it is Bentham and Hooker or Theophrastus or probably anybody else, or even the present APG, in fact, philosophically, principally.
Sir, yes. Sorry to interrupt you, sir. Some people are saying that they cannot see the screen. So
no, but it is visible to you, no, right? Is it visible?
Yes, sir. I can see from my mobile, sir. So let us resolve that, sir, because some more people are responding on that.
Yeah, tell me how to go about this.
Sir, how about Vijay sir and madam? Are you able to see sir's PPT, madam? Sadhana?
Yes, Rijan. It is seen.
Madam, I want a response from participants too. Who can see, they can just message me. Like they can see or they can't see. So for some people it is visible. So I request those people who cannot see, just go back and rejoin again. Some are saying that we can see, we are not, we cannot see. So there might be some problem, technical error from your side. I request who cannot see the PPT can just go back and come.
Yeah, we can wait for another 2 or 3 minutes to start out this issue.
Yes, sir. Yes.
Yeah.
And dear friends, this is also visible on YouTube. There is no problem from the speaker side or from our host side. Sir, on which slide are you there, sir? On first slide or on
first
second?
First slide, sir.
Only first slide, right, sir? Yes, sir.
First slide.
So they are confused, sir. There might be no problem. We can proceed, sir. Thank you.
Okay, right. So as I told you, all the classifications that is there in the history are all in a way artificial. If you consider based on principles and philosophy, every classification is human made. Whatever that is human made, it is definitely artificial. There is nothing called artificial, natural, and other kinds of classifications. So how do we understand these classifications? For me, there are only two kinds of classification. This is the current way of looking at classifications. One is phonetic, the other one is phylogenetic. What is phonetic? There is more noise at the background. The phonetic classification is based on overall similarities. There is no phylogeny inbuilt into this. Particularly Bentham and Hooker, of course, as we have all known this system very well in India, this classification is absolutely a phonetic classification. Phylogenetic, which of course, after the Darwin's theory, all the systems like England, Prandtl, Tractajon, Cronquist, and the current APG, everything is based on phylogeny, and they are all phylogenetic systems. So all classification systems can be put under two categories. One is phonetic, the other one is phylogenetic. So we can conveniently forget, and let's not use these terms like artificial, natural, and phylogenetic in our syllabi hereafter. Let's talk about only phonetic and phylogenetic so that we are talking in terms of actual meaning of these terms. Otherwise, we are putting our students unnecessarily into confusion. What is artificial? What is natural? So everything is natural and everything is artificial in a way. Right. Let's go into the core subject now. Of course, the history of classification starts from Theophrastus. He's also called father of botany. And why, of course, every history, not only the classification history, every history starts from the Greek land. So, because that's where the history is recorded. Elsewhere, the history is not recorded so clearly. Later on, of course, other people started recording it, but the first records of anything, whether it is medicine or probably whether it is botany or zoology or even philosophy, anything starts from Greeks. So that's why, of course, we always have the Greek people coming into as forerunners in the history. And Theophrastus, you know, is, of course, in those days, there is no subject called botany. They're all philosophers, but the philosophy included also science. And that's how, of course, they are interested in the plants and they started looking into the plant kingdom. Two works by Theophrastus. One is the Inquiry into Plants and the Causes of Plants, and of course the other one, the Historia Plantarum. These are the three books by Theophrastus, which talks about the classification of plants. And this is, of course, he talked about 480 plants in his book.
Sir,
yes,
excuse me.
Please.
Slides are not moving.
No, I'm not moving the slides. I'm talking only based on one slide now.
That's fine. That's fine.
So it's not right because there is
sir,
more information.
Sir, if you don't mind, once can you show the second slide and come back, sir? Because the participants are continuously asking.
Sure, this is the second slide. Right? Okay.
We can't see, sir.
No, sir, not moving.
This is the second slide. Can you move? See, I'm moving the slides now.
No, sir. We can't.
Now?
No, sir.
No, sir. Kindly stop your sharing and can you share again, sir?
Sure. Yeah. Is it okay? Can you see?
Yes, sir.
Sir, we can see, sir, but we have to get the response from the audience again. Sir, go for slideshow, sir.
Next.
Participants, please respond in the chat box. Those who were unable to see earlier, can you see again?
Yes. Yes.
Yes, sir. Done, sir. Thank you, sir.
Done? Okay, right. Thank you.
Yes.
So, Theophrastus, of course, is a Greek philosopher. And most of the earlier classifications, up to a certain period, they were all habit-based. Like if you go into the tribal areas and ask them to classify, they will simply say, these are the trees and these are the shrubs or herbs and other things as climbers. Sometimes they may not have a term for certain groups of plants, certain habits of plants. So this is also in a way natural way of looking at things. And that's why most common people also classify. And but Theophrastus, more than that, this kind of habit-based classification, he has used certain characters in his classification, like determinate and indeterminate inflorescences, which we still use, like the racemose and cymose inflorescences, and the position of the ovary, hypogyny, perigyny, and epigyny. And of course, the very popular way of looking at the system is the union of petals. And that's why, of course, we have been learning Benthamenocal system, polypetalous and gammapetalous corollas. Then another thing is monocot and dicot, which started in 3rd century BC. So we are still probably continuing on some of these things. So we have not deviated much. Then, as there are a number of Theophrastus disciples who are in the scenario, I am not going to probably tell you about everybody, including Aristotle. The Pliny, of course, next person is Pliny, who wrote a book called Historia Naturalis. And basically, of course, he did not differ much from what Theophrastus has discussed in terms of classification. But a major change that has come in the taxonomy is at the 6th century AD by a person called Dioscorides. And he made a very important contribution to taxonomy. That means he shifted from looking at from the habit-based classification, he shifted to the utility-based classification. That's a major perspective change that has happened. And from then on, it was the uses of plants that dictated the classification, particularly the medicinal uses. So Dioscorides wrote a book called De Materia Medica. And this is, of course, it's available in the net. You can freely download and see. There are more than 600 species of medicinal plants that were listed in this. But I'm going back to the earlier slide. What is more important about this book is it's an illustrated book. In fact, first illustrated flora, we can call this as a kind of a flora. First illustrated flora started much, much early during the 1st century AD. So this is the major turning point for the publications that followed after Dioscorides. And plus Dioscorides' book, illustrated book, continued to be the only source of reference for nearly 10 centuries in Europe, which is called Dark Ages of Europe. So, and there is no other book. And this book was meticulously copied, handwritten, and all the drawings which he made are probably wood carvings, and these have been reprinted again and again. You can imagine the kind of inputs, the kind of pains that people have taken to create a copy of Dioscorides. Today, of course, it's much easier. So that was the tremendous influence of Dioscorides' book in the entire learning of taxonomy. It is based on only medicinal plants. And in those days, medicinal plants are going to be very, very important for people. And the idea of learning taxonomy itself is to identify medicinal plants. Then came, of course, Herbalist Period. I will leave, of course, a few people. It's you can probably really see from the side and note it down. The Herbalist Period starts from Dioscorides. Herbalists are people who are medicinal plant specialists, are probably who practice medicine. Today, we also have herbalists in like Vaidyas, what we call as Vaidyas. And in those days, they are called herbalists in the Greek world. So there are a number of herbalists who contributed to taxonomy, and their focus is only medicinal plants. One of the well-known herbalists, of course, who gave a good contribution is Otto Brunfels. And Otto Brunfels is from 15th century. He has written 2 books, Herbarium Vivae Icons. Icons, of course, is the illustrations, which is otherwise illustration of medicinal plants. So he recognized 2 important factors which we use today for plant classification. One, perfecti and imperfecti flowers, that is, flowers with full complement of organs and flowers with incomplete complement of organs. And he also recognized certain groups of plants, such as Lamiaceae and things like that. And following, of course, Brunfels, there are several kind of herbalists who participated in the taxonomy history. All of them, of course, contributed a little more in understanding the plants, and also probably including more plants from Europe and other parts of India, other parts of the world, particularly from India, because in those days we had a big connection, especially trade connection from India to other parts of the world. So we were trading with Rome, we were trading with Egypt, we were trading with other places. So that is what, of course, carried plants from India and surrounding regions to the Europe, which were recognized by people there. So that all these things were happening. And Leonard Fuchs is another herbalist who followed. Matthioli is another herbalist. Clusius, Gerald, all these people have contributed during the herbalist period. And their contributions, though not very significant, but still they contributed something to the plant taxonomy. But if you look at the names, you can easily figure out those who, of course, who study taxonomy as their main branch of specialization. They can easily understand these names are commemorative names these days. Brunfelsia is a genus, Fuchsia is a genus, Clusiaceae is a family, plus Clusia is also a genus. So these are commemorative names. And the first taxonomist to appear in the history is an Italian botanist called Andrea Cesalpini. You can also see this name is also a commemorative name in taxonomy. You have a family in his name, you have a genus in his name. And he classified plants based on definite morphological features. Fruits, seeds, and embryo are characters. And color of the sap is another character. Number of ovules in ovary is another character, which we still use in your practical classes. Right? The kinds of fruits, the kinds of seeds, and of course embryos, oh, we don't use much, but still it's a very useful feature of classification. Number of ovules, of course, you still use. So these are features that have a history in their appearance in taxonomy. Duplantis is another botanist who described about 1,520 species, both woody and herbaceous. And for him, he relied flowers and fruits as the most reliable characters. And that's what we still practice. And if anybody brings you a branch without flower or fruit, either a research scholar or a student would say, no, I cannot identify this because there are no flowers and fruits, immediately turns away and send them. And this habit is probably a very old habit, right from the days of Duplantis, who relied on flowers and fruits for identification. Jean Bahin and Gaspard Bahin, these are called Bahin brothers. And that's why you have a genus called Bahinia. Bahinia is a plant with kind of two leaflets attached together. So you can very well understand when you see the leaf. These brothers, they were always together, they worked together, and of course several things they published together, though they had their own publications. And Historia Plantarum Universalis is one of the major book that these brothers have published. They've described about 5,000 species. And as I told you earlier, Dioscorides set a pattern for illustrated floras, and that continued for a long time. So this is also an illustrated, which made why illustrated floras were needed in those days is it was easy for people to identify plants. They don't need a description, they don't need the dissections. So they can simply look at the diagram and identify plants. So that is why they were much preferred and everybody was making illustrated floras. Today, of course, they are becoming much more popular through photo guides. And as I go through the slides, if you are looking at the numbers carefully, you see the numbers are increasing. At every point of time, at every point of history, the plants that are known to botanists and taxonomists are increasing. We started with Theophrastus with 480 plants. Then it came to 600, then went up to 1,520. Now it's of course 5,000 species. And when Gaspard Bahin wrote Phytopinax and Pinax theatri botanici, he recognized 6,000 species, another 1,000 species more. So, and the contribution of Gaspard Bahen is a major contribution. He recognized the kind of generic and species concept. So species concept per se and is somebody else, but still in a kind of a rudimentary form, they recognize generic and species concepts. Tournefort is the person who was regarded as father of generic concept. He defined the genera so very clearly, which still probably are valid, which still use many of his genera. And he wrote a book called Elementary de Botanique and Institutionis Rei Herbaria. And all in all, he described 698 genera and 10,146 species. So you can see a quantum jump in number of plants that is known to botanists in Europe. And he provided very good generic descriptions. Many of his genera are validated by Linnaeus. And these genera, of course, are still in use. For example, Abutilon, Quercus. These are still in use. So that means, imagine the kind of solid base the Tournefort has provided for generic concept. A sudden change has happened in the history of classification. This is probably a second scenario. The first scenario is either habit-based or probably overall kind of similarities. But when Camerarius, Rudolf Jacob Camerarius, when he discovered sexuality in plants and also experimentally proved it, and this changed the entire idea of classification. And this probably set a kind of a platform for a shift in the classification mode and for Linnaeus to come in. And that's why we call Linnaeus, though we sometimes, of course, pronounce these names very differently. Carl Linnaeus, in fact, he is much known as Linny in his own country, Sweden. He's, of course, if you go to Sweden and say Linnaeus, they may not understand. It's typical English pronunciation. So Carl Linny or Carl Linnaeus, his system is well known. This is where we call sometimes these systems as artificial systems. So they are not, of course, truly artificial. They are not, of course, every system is artificial, I told you as earlier. And he published his first paper on sexuality of plants after Camerarius. And he wrote several books. Some of his books are still very useful for the rules of nomenclature and the naming of plants, because he has given everything in as early as in the 18th century. And some of these books, especially the Systema Naturae, which is a kind of philosophy for classification, where he developed guidelines and rules for classification, and everything is connected to taxonomy. And so these are the books that are foundations for classification. And this Genera Plantarum, Linnaeus Genera Plantarum, describes about 935 genera, and there are 5 parts or 5 editions which they have published, and there are 336 genera grouped in 24 classes. And these classes, and in all, he described about 5,000 species in 1,000 genera, established a binomial system. That is Linnaeus' greatest classification. Unfortunately, in history of taxonomy or in history of botany, we reduce Linnaeus to this. If you ask any of the botany student, tell me something about Linnaeus, he will immediately say he is the father of binomial system. No, he's much more. Linnaeus is one person, of course, who's a typical taxonomy person who classified anything and everything. In fact, he was much wanted in Europe to classify many things. There are lots of princes and kings who called him to classify their plants in the garden, or their objects they collected, the ornaments that they have, or the precious stones which they have in their museums. Everything can be classified by Linnaeus. That's why in Sweden there is a saying, God creates and Linnaeus classifies. And that was the pride that the Swedish have about Linnaeus. So Linnaeus is much more knowledgeable on several things. Linnaeus is also a kind of a first ethnobotanist. So when people read ethnobotany, history of ethnobotany, people forget about Linnaeus. In fact, he is not one of the, he is the first ethnobotanist who studied tribes of Sweden who lives in the area called Hartus Upland, the Uplandica. The upland is a hilly area in Sweden. He went there, he stayed there for a few years, studied the use of plants by the tribes there, and then came back. So he's probably the first and foremost ethnobotanist in the history. So Linnaeus has several credits. Apart from that, it is because of Linnaeus, though today Greek is out of Greek and Latin, of course, particularly Latin is out of scene in plant taxonomy. We have brought in English for much wider uses. But still, at least 4 or 5 years ago, Latin was much needed, at least for the diagnosis of plants, new species and new groups, and Latin descriptions were needed. But before that, everything was in Latin. That's why, of course, people say botany is Greek and Latin. And Linnaeus modified the very classical Greek Latin to suit the botanical descriptions and the botanical usage. That's one of the another greatest contribution which Linnaeus has given to the system of classifications, to the taxonomy. And these are well known. These are there in every book of our traditional textbooks of taxonomy, all the 24 classes. If you look this, it's very simple, right? Of course, his classification is based on, there are 2 or 3 things. One, number of stamens. That's why starting from 1, monandria, diandria, it goes up to dodecandria, which is 12. And length of stamens, whether stamens have similar lengths or different lengths. And the differential length of stamens, whether it's didynamous or tetradynamous. And union of stamens, whether it's monodelphous or diadelphous, and arsinginaceous. And union of stamens with the ovary, whether it is they are united with the gynoecium. And how are they placed, whether they are placed the same flowers or in different flowers. And all these are his classes, and very simple, and it served the purpose for many of the taxonomists in those days to identify plants. If it is 2 stamens, you should look into diandria. If it is 3 stamens, you should look into triandria. So people found it very easy, and that's how they followed it for at least several decades during his period. And this book was irreplaceable until the next scenario changed the classification systems. So Linnaeus was a leader in classification. And those who argue, of course, this is where I stress my point, those who argue Bentham-Manucher system is very easy, and we are used to this. During Linnaeus period and after Linnaeus period, this system was easy for them. Look at how easy it is. Just 1, 2, 3. Just see the flower, see 1 or 2 or 3, and go into the class and identify your plant. But people have changed. People have come to other systems. So we cannot be sticking on to one system. If Bentham-Manucher is easy for us, for many of the Europeans in those days, this was easy for them. But they've changed. Then, if you keep an eye on the characters and the kind of groupings people have made, you can easily understand. We slowly get connected to the systems of classification, particularly our Bentham-Manucher, through these histories. How things have been changing, how things have been evolving to a fine and perfect system of classification, which came later during the Bentham-Manucher period, which is a typical phonetic classification. All these systems of classification are phonetic systems of classification. And if you look at next few people, the Juseyu family, because in those days, people studied as families. Even Linnaeus' son was a taxonomist. So that's why sometimes you find LF as an author. F means filius. Filius means son. So this LF, and there are lots of Fs that you can write, hook and hook F. So father and son, father, son, and of course his grandson. So a lot of people studied as a family the botany, and particularly the taxonomy, because it runs into their mind, it runs into their blood, it runs into the family, and there are already enough kind of literature that is available in the family. So Juseus have a great influence in the history of taxonomy. The classification published by Anton Laurent Juseyu, Genera Plantarum. So here another thing which I want to probably remind you, the Genera Plantarum as a common title was used by many botanists. Even Bentham and Hooker also used this. So if you say Genera Plantarum, you cannot imagine an author and say to someone else, if you want to be specific, you should say whose Genera Plantarum you are talking about, whether it is Linnaeus Genera Plantarum, whether it is Jusseius Genera Plantarum, or Willdeno's Genera Plantarum, or probably Bentham and Hooker's Genera Plantarum. So you should be very specific about authors. If you simply say Genera Plantarum, it is a very broad kind of a title. It could mean anybody's Genera Plantarum. So we should be very, very careful in handling some of these things, very, very careful in probably in telling some of these things. We cannot be vague in telling, particularly taxonomy requires specific ways of telling. Okay. So his classification, that is Jusseiu classification, classified plants into two groups, which we still use it. Acotyledonnae, uncotyledonnae. Those which have cotyledons, those which doesn't have cotyledons. Very simple. And the cotyledonous flora are classified into two groups, monocots and the dicots, though he did not use the term dicot, which are again probably are classified into three groups: apetalae, those doesn't have petals; monopetalae, those who have single word of petals; and polypetalae, those who have many words of petals. Can you see a kind of rudimentary system of classification of Bentham and Hooker into this? If you overlay Bentham and Hooker system, you see it fits into perfectly, if not with every aspect, but it fits into some of these groups. John Ray. John Ray is regarded as a father of species concept. If probably we recognize species in such precision today, we should thank John Ray for that. He's an English botanist. Similarly, if you also probably can, this is probably is another bit of information which I like to share with you. The dominance in terms of taxonomy, because this is the field that contributed not only to the science, also to the wealth of the nation. So that's why each country was promoting their botanist. And the history of taxonomy will be shifting from one country to another country until, of course, British took the supremacy in this subject. If you look, it may be from Sweden, of course, it emanated from Greek, Sweden, then of course French, then other botanists. So, and now English botanists are coming into, John Ray. So John Ray, he wrote two books, Methodus Plantarum and Historia Plantarum. And one of his dictum, which he followed in the classification, is all parts should be given equal importance. Of course, later on, this also came to be known as Adamsonian principle. And the Adamson, of course, on whose name you have Adamsonia, who promulgated that all characters are equal important and every character should be given equal weightage in classification. But unfortunately, we still follow the earlier dictum that fruits and flowers are going to be more important characters. So, and that's what we still follow in our classification system. We have forgotten some of these kinds of early rules. And in total, John Ray has studied about 18,000 species, which is another quantum jump in the numbers. And look at his classification. Again, there is again a kind of rudimentary Bentham and Hooker system of classification. And here, of course, he divides plants into two groups: herbs and trees, herbae and arboris. So herbs, herbae means herbs, arboris means woody things. That's why we call where trees are planted as arboretum. The herbs are again classified into two things: florae destitutae, that is plants without flowers, which are of course cryptogams, and fructifications, herbae fructification, that is plants with flowers. And again, he had two kinds of things, dicots and monocots, which are well known to you. The arboris, again the same thing, florae fructo remoti, that means flowers that are very well visible, and florae fructi contiguous, that is, which are well visible. Major person who was contributed, major person who has come into the taxonomy, who has contributed enormously, and whose books are still referred, because there is no replacement for many of his the family treatments, which treated the families as in a monographic way. There are, of course, other people who later also have given such a kind of conspectus. Augustine Pyramid de Candolle. He wrote one of his famous book, though there are quite a few publications. One of his famous book is this, Prodromus Systematis Naturalis Regni Vegetabilis, very popularly known as by the first word, Prodromus. So this Prodromus is 17-volume book. Imagine the kind of, because imagine this is a single person who's writing this one. And 17 volumes written, which is consisting of 58,975 species. The kind of enormous understanding of these people comes from looking at the diversity of plants and knowing each and every plant that they have described in their book. That is, it's a personal knowledge, and it's also a kind of elaborate knowledge each one of us have. But look at the present conditions, we are unable to dissect and study even 20 or 30 families, and of course, try to describe these plants. So we have shrunk in our exercises, and that's why we fail. The taxonomy is very difficult. But if de Candolle has probably thought that way, he would have written 17 volumes and 58,975 species described. So his classification is also, again, a kind of precursor to Bentham and Hooker classification. The plants, of course, generally the kingdom Plantae, which of course he described into the vasculates, which we call Tracheophyta today. So you can see how terminologies are emerging. And cellulars, which are probably the non-Tracheophyta without the vascular tissues. And again, this exogene and endogene, right? So exogene and endogene are plants without the kind of with flowers, and diploclamidae and monoclamidae. Diplo means 2 words and mono means 1 word. So it is, you can see the kind of monoclamidae and the polypetalae, gamma-petalae concept emerging here. And the endogene, the phanerogame, which is the flowering plants, and cryptogame, which is the non-flowering plants, right? So this, of course, the exogene, endogene, in fact, are another terms which are equivalent to our current understanding of monocots and dicots. The cellulars, which are not vascular plants, non-vascular plants, they are into there are 2 things, foliaceae and aphillae, of course, which we have no equivalence of these terms today. But all the thalloid or foliaceous thallus are into foliaceae, and structures which are not like this, maybe filamentous and other kinds of things, are included in this kind of thing. And if you look at the earlier classifications, another feature which you should be noting down, or which you should be understanding by going through this kind of classes. They've not only classified the flowering plants, they've classified the entire plant world. There are fungi in it, there are algae in it, there are pteridophytes in it, there are lots of things in it. So this is something which is a feature of earlier classifications, which of course are missing in today's classifications. We don't have this kind of a comprehensive system of classification because the volume of knowledge in each field has increased tremendously. So it is not easy for one person to understand every group and probably try to classify. Next comes our heroes, George Bentham and Joseph Dalton Hooker. Because I call them as heroes, particularly in the Indian scenario of botany. They're still heroes. And they probably are not replaced, though every part of the world have replaced their systems of classification, but we are still clinging on to this system. As I told you, they wrote Genera Plantarum. This is again another general title, but if you say it is Bentham and Hooker Genera Plantarum, it is not simply Genera Plantarum. Written in 3 volumes. I don't know how many of you have seen this Genera Plantarum, a wonderful treat. In fact, as MSc students, we went and searched for this book, we found in some of the libraries the old volumes, and whether we understood or not, at least we want to see the book and just turn the pages and see, because everything was in Latin. Though we can understand few terms and few things rudimentarily or probably in a preliminary way, but it is not possible for us to read pages and pages of Latin information. But it's a kind of an experience for us to see some of these classical books. And this system is essentially based on decondole system. So this, all in all, he described about 97,205 seed plants. We call it a seed plants because they included also gymnosperms. And that's suddenly, of course, we say, what are the merits and demerits of this? Is one of the very standard question in our taxonomy papers. Not only standard, a very historical question, keeps appearing in every examination, which I feel is a very not a right way of saying. In fact, I would say it's a nonsense question also, because there is no demerit for any classification. Every classification is evolved in a particular circumstance with a set of particular knowledge and in a particular period of time. So you cannot call it as a demerit, right? It's like calling bullock cart as a demerit vehicle and probably the aeroplane as a merit vehicle. So you cannot compare the two different periods and two different probably the lifestyles and several things during those periods. But unfortunately, we started using these words in our systems of classification in our question papers, merits and demerits. At least hereafter, I would request teachers, please avoid using these terms. If you say, you can say, what are the major, probably don't say demerits, what are the criticisms against this classification? Because criticism evolved later. So any system, when it is built, nobody can build it with a demerit. So, and this is a kind of our viewpoint, in fact, our biased viewpoint that we put on many of these systems. So kindly avoid some of these terminologies, merits and demerits, use criticisms. So that probably you are also telling your students not to be biased about one system, or probably not to be taken granted for one system. So you have to be very scientific and critical. That's the kind of mental ability that we should be building in our students. So this system, I'm not going to explain to you in a detailed way, because all of you know, all of you study right from your school days up to probably MSc, even if you have a probably paper in PhD, that is also probably only based on Bentham and Hooker. So this is our lifetime classification, I would say. And this classification is as well known, but on what basis this classification has been built? Because apart from well-known dicards and monocards, in between which, of course, they place gymnosperms, it is based on a few major principles. That is, one principle is the more the parts, the lesser or probably the less developed is the plant. That's why we put Polypetales, Renalis, Ananaceae in the first. The fewer the parts, the more advanced is the group. And when the parts are probably free, and today, of course, we don't use the word free. There is subtle differences between free and probably distinct. So the terminology is also changing. What we use today, of course, instead of Polypetale, though it's Bentham and Hooker who use Polypetale, we use today Apopetale. Apopetales condition or probably Sympetales condition. The Polypetale, there is union of parts. If the floral parts are too many and probably free or apo, they are probably towards primitive. And if they are fewer and if they are united, they are towards the advanced. That is one of the principles which Bentham and Hooker followed. That's what you see starting from the Magnoliaceae to probably Umbelliferae and other things in Polypetale, and from, of course, Rubiaceae to other families, the Gamopetale. But another thing that they followed is also the position of ovary, which is well reflected in earlier classifications. Remember, the positions of ovary like inferior, superior, and semi-inferior have already been used in the classification. have already been recognized for the purpose of classification. So Benjamin Hooker also used this character for classifying plants. If you look at, he puts the superior ovaries as towards the primitive condition, and half sunken or sunken ovaries into in-between category, and fully inferior ovaries into the kind of the advanced category. So that is what is being followed in Polypetalae. And in Gamma Petalae, of course, it is a bit of reverse. You see Rubiaceae and Asteraceae with inferior ovaries in the beginning. And later on, of course, if you see based on the position of ovaries, Asteraceae is one of the most advanced families, which of course all of you would have known and studied. So these are some of the basic principles which he followed. Otherwise, from complex to simple, that is one theory that they followed. Complex structure of flowers and to a simple structure of flowers, which means complex to simple is the advancement. So that is the philosophy which they followed, and that's what you see in the Bentham and Hooker system of classification. Again, what changed? Just like the discovery of sexuality by Camerarius, which changed the entire idea of thinking, especially for Linnaeus, the Charles Darwin's book, which was published in 1858, just before, probably almost, if you look at the Bentham and Hooker, this is published between 1862 and 1883, right? So it is just before the Bentham and Hooker's publication. And all of you know the book Origin of Species. I don't know how many of you read the book. Today, of course, cheaper editions have come. And this book has totally changed the way that we looked at the biology, the world of biology, not only classification, the entire world of biology. In fact, there is a saying, if you're not looking through the glass of evolution, your biology is something that is very primitive. So that is the kind of influence which Charles Darwin, Origin of Species, has brought in. Everything is being looked into from the point of evolution and from the point of phylogeny. And yes, the Charles Darwin, based on Charles Darwin, there are two schools, of course, which will come a little later about this. So from Charles Darwin, the phylogenetic system, the so-called phylogenetic systems begin, because he has built the idea of phylogeny or the evolution into the classification. And the first kind of thing that came into being is Engler's school, from Engler's school in 1859. It is not Engler. First to put forward and use this kind of a theory is Alexander Braun. And everything emerges when you are working on a group of plants. And Alexander Braun was working on the flora of Brandenburg, which is a kind of a district in Germany. And for him, he thought in reverse fashion, what of course the Bentham and Hooker have thought, from complex to simple. Alexander Braun thought simple structures are most primitive structures, and advanced structures have most evolved structures, complex structures. So that is their idea about the evolution. That's why they put, if you look at the so-called dicard, they put apetalae, that is like catkin, things with catkin inflorescences, like for example, your Casuarinaceae, Piperaceae, and all those things as most primitive kinds of things. And then go on to elaborate flower structure, Leguminaceae, which is a very beautiful structure of flower with definite parts and also with specialized parts. So that is their idea about the classification, which moved from simple to complex in a reverse fashion, what is practiced by Bentham and Hooker from complex to simple. And unfortunately, this idea of classification is very short-lived, not followed by many people. The other person who followed is Eichler. And Eichler also, of course, has written quite a few books in Germany. And you can see the focus from English now is also shifting to German botanists. So he followed the same pattern which Alexander Braun has followed. And this is his classification, though we will not be going into details of this classification for want of time. And you can see, if you quickly go through the structure, you can see how it follows the idea of simple to complex. This, of course, is probably one of the well-known classification of the Braun's idea. Engler and Prantl, both are German people, and their book is known as Die Naturlichen Pflanzenfamilien, and much popularly known as Pflanzenfamilien. Like Prodromus, this Pflanzenfamilien is another very unique book, because still many families are treated in a monographic way only in this book. We don't have a monographic treatment for many of the families in the world other than this book. So of course, now there are things emerging, but still this is one of the most comprehensive treatment for the entire world. And here also you see, following the traditional pattern of classification, they've classified the entire plant world from algae to flowering plants, whether it's gymnosperms or anything else. And here also you can see they followed from simple to complex, from lemnales, of course, starting from here, though it is not written there. Microscopic, which is Orchidaceae. Orchidaceae is the most complex structure. Lemnae is a very simple structure. And the siphon and the dicots here, right, from sympatale, and according to that, from simple structures to complex structure, campanulate, other things. Here also, of course, simple to complex. This is their overall system of classification. If you look at overall, they have classified about 303 families. Right? 303 families they have defined and classified. And his system, the Bessie's system, is an English botanist. Bessie's system is well known by the way he represented, because he put the ordering of groups one above the other, like a structure of a cacti. That's why Bessie's cacti is a well-known phylogeny kind of representation. In fact, I would say he's one of the first person to draw a kind of a phylogenetic tree, though it emerged in the form of a cacti. This is probably one of the first known phylogenetic tree in the history of classification, especially in the phylogenetic classification systems. So Bessie, of course, he classified things again based on what was known from simple to complex, and that is Bessie. And Bessie's cactus is well known. I don't have a Bessie's cactus picture. Hutchinson, again an English botanist, who is well known, of course, who is well respected even today for the kind of circumscription which he has given for families. Hutchinson families are well defined, and together, of course, he has in his book, Families of Flowering Plants, he has described about 418 families. And this is a treat to read. If you see Families of Flowering Plants, he makes it easy for you to understand families, where he has given the kind of characters that classify the family are probably so-called, today we call it as diagnostic characters or important characters of the family, which he has given it in his book in bold letters. Those who read Families of Flowering Plants will understand this. And in fact, these books set a kind of a major information system for the later evolutionary systems to evolve. And Hutchinson followed very, in a way, though it is a evolutionary system, phylogenetic system, he followed one of the primitive way of looking at things. That is, he divided the entire plant kingdom, particularly dicots, into two things. One is lignosae and the other one is herbaceous. That is woody plants and herbaceous plants. This is a very basic, that's what Theophrastus did. Probably that's what earlier people did. But later people have changed. But Hutchinson reverted back to this system for some of his personal reasons. And this is, of course, this has attracted a lot of criticism among botanists. But whatever may be the criticisms against this classification, but this is one of the classification that has circumscribed the families very clearly, and his families, probably some of his families, are well known today, very well known today, are still followed today, and in fact also paved way for our understanding of the complexity of the families. Takhtajan is a Russian botanist. This, of course, first time Russia is coming into the picture in the history of classification, but this made revolution in the history of taxonomy. Takhtajan was less known in the field of taxonomy for quite a long time until his book was translated into English. The moment it was translated to English, this book became immensely popular among all European countries, and they started adopting the terminology followed by Takhtajan. So far, we have seen different kinds of terminologies in earlier classifications, but now the Takhtajan classification probably put forwards a totally different terminology. There are no dicards and there are no monocard terminology that is being used. He recognized entire plant kingdom as Magnoliophyta. This is a term that is used. Entire plant kingdom as Magnoliophyta, and the dicards as Magnoliopsida, and the monocards as Liliopsida. And these terms have followed later by several phylogenetic botanists or evolutionary taxonomists, including Conquest, and of course, to some extent, the latest Kubitsky, or earlier Dahlgren. Many of these people have followed these terminologies, Magnoliopsida and Liliopsida. That means replacing the words like dicards and monocards, which of course puts in terms of a different perspective, right? You have a group which characterizes a larger group, Magnolia or Magnoliaceae, that characterizes the entire Magnoliopsida, and Liliaceae that characterizes the entire Liliopsida. Again, Magnoliopsida is divided into several orders. Magnolidae, of course, there are different terminologies that are given. Ranunculata, there's a spelling mistake. So all these things are subdivisions of Magnoliopsida, and similarly Liliopsida is divided into 4 subgroups. The Cronquist, who simply adopted Takhtajan's classification in English, and because he's an English botanist, the classification became immensely probably adopted by several people in the Europe. And if you look at the basic classification, it's simply just superimposed on the Cronquist classification, except one change he made. Instead of looking at 4 groups, he looked at 5 groups in Liliopsida. That's the only change that he has made. Otherwise, all the rest are the same and in concordance with the Takhtajan. We are now going into a different phase of taxonomy. Most of you would have studied a chapter on numerical taxonomy in your syllabi, whether it is a BSc or MSc. This is again something which is a very traditional question that appears, and I don't know how how much you have understood about the numerical approach. And this approach, of course, has begun from a very classical textbook written by 2 people who are not actually biologists, Sneath and Sokal, called Numerical Taxonomy. And it is this book that set path for the entire numerical approach to taxonomy. What do we mean by numerical approach? In all the earlier classifications, we were using many characters and different variations characters. We use leaves, we use flowers, we use fruits, we use seeds, all kinds of things. But they have been recognized only by the terminologies by which they are known. But when it comes to numerical approach, it is a completely computer-based approach. And the computers will never understand because it's a machine language that you have to use. A machine language cannot be descriptive. You cannot say this is ovate leaf, this is lanceolate leaf, or probably this is apopetalous flower, this is sympatalous flower, all kinds of terminologies. Your machines will never understand. So machines need to understand only through digitization. You have to digitize the characters. So that's when the numerical approach to taxonomy has begun. That is, the advent of computers have brought in this approach. This system is probably hailed in the beginning as something that is unbiased. And unbiased in the sense, you're not trying to influence this. Trying to look at characters, okay, this is more important character, this is not, probably this is a very trivial character, we will not pay much attention, or this may not be used in classification, this may not be useful in classification, it's a variable character, all kinds of things we have been using, and it's a bias in classifications. But this numerical approach, when you give a numerical codes to characters, 1, 2, 3, 4, whatever it is, so there is no bias. 1 means 1, 2 means 2, and you feed into a computer, it classifies based on whatever that you feed. So absolutely the bias is brought to nil. And that is why this classification has suddenly become a very important way of approach. And the numerical approach has branched into two. As I said earlier, the phonetics, the book that is by Smith and Sokal practices only phonetics. That is by overall similarities. And that's what we have been probably working as numerical taxonomy. If you go into your library, you may have several dissertations done by your MSc students, MPhil students in numerical taxonomy. Particularly, of course, I know Andhra Pradesh, a large number of students jumped into this kind of studies. So that is simply a phonetic way of looking at it. But in 1960s, a different branch or a different way of looking at, a different way of analyzing has emerged. That is what is known as cladistics. So what is these two? What defines phonetic and what defines cladistics? If you look at, they differ mainly in two characters. Of course, leave alone this is only a representation. This is only a terminology difference. But basically, they differ from two ways. One is the phonetic, it gives equal weightage to both primitive and advanced characters. It doesn't distinguish whether it is a character is primitive or advanced. It simply compares every character in a general way. Whereas cladistics gives the weightage only to advanced characters. It doesn't even take into consideration the primitive characters. So that's one of the major difference between phonetic and cladistic approach. Another phonetic and difference between phonetic and cladistics is it treats both analogous and homologous features equally. All of you know what is analogous and homologous features. Of course, I'll explain a little later for the benefit of students. And in cladistics, only homologous features are considered. Analogous features are not at all considered. So that is the major difference between phonetic and cladistics. Otherwise, there is a kind of philosophical difference, the way it is looked at and the way it takes into weightage for analysis. So, and if you understand this is the basics for today's phylogenetic programs and today's APG classification. If you understand this basic, rest of the things are much easier, right? And today's classifications, particularly all the phylogenetic classifications, whatever that is appearing in the journals and other publications, they're all based on cladistic analysis. So cladistics is something which is a computer kind of a program that is used to analyze the characters. And it is particularly built in to analyze using these two things, an advanced character and homologous character. So that, and entire analysis based on this philosophy. And that's why you have several changes that are happening. And what is this cladistics? Is this cladistics completely a kind of a taxonomy based tool, or is it a kind of a general computer tool? This method was first developed by Willy Hennig, a German entomologist, when he was trying to classify insects. And this has beginnings in 1950s. Right? Whereas in other parts of the world, it spread so quickly, and to descend into our syllabi, to get into our minds, and of course, to probably get into our practice, it took nearly 60 years for us to learn this and to get into the practice. So we are very late runners in this field. Cladistic methods, they can be used to group any comparable data into sets and subsets known as clades. So that's why it's called cladistics. Clade means a branch. What you see in a cladogram are branches, and these branches are probably denote the groups that are similar and the groups that have emanated from one ancestor. So that's why it's ancestral clades. So these clades, and that's why in cladistic systems also, we don't use probably terminologies like order, suborder, family thing. It's only clades. So we use the word family, but above we don't use these words. We use only clades. So any set of data in the sense, now of course people have compared languages in the world and have grouped them into different groups. You have the Dravidian group of languages, to which of course Telugu and Tamil and Kannada belongs to. You have Indo-Aryan group of languages, to which Sanskrit and German and other kinds of European languages, of Indo-European or Indo-European group of languages, to which other language, the Sanskrit and European languages belong to, because all of them have similar semantic structure. And this is possible because through cladistic methods, we analyze the language structures, and it is possible to group and develop a cladogram and how these languages are grouped. Similarly, now anything can be grouped, including your Carnatic ragas can be grouped based on cladistic analysis. Which are the ragas that are closely similar, which are the ragas that are different, how ragas probably have evolved from one from another. So all these things are possible using cladistic methods. And today the so-called bioinformatics extensively uses the cladistic programs, especially to design drugs. The drug design is something that is dependent on the cladistic programs, which are specially built for certain purposes. So cladistic is something, it's a tool which can be used for anything. Of course, we use it here to define monophyletic groups in the classification. And what do you mean by, how do you, of course, understand the cladistics and the phonetics they work? See, for example, I try to explain to you with a set of 3 taxa and 5 characters. And as I told you, the characters are coded. Characters are numerically coded. Here we have used only 2 codes, that is 0 and 1. It could be anything, broadly classifying absent or present, right? So, or probably this or that. So that's all. No further classification here, because it's for the ease of explaining. We have just used binary code of coding. That means only into 2 states, so 0 and 1. So 3 taxa here. If you go by a phonetic method, that is overall comparison, which many of the classification system, which probably we ran through the history, they've all used this phonetic system by classifying overall similarities. If you go by overall similarity here, how many zeros are common between A and B? 3 zeros. So 5 out of 3 characters are common between A and B, which means they share more number of common characters than C. C does not share any common character. Automatically, if you use phonetic principle, of course, that's something that is also a kind of a general thing that is there in our mind. We compare overall and then say, this is much more similar to this. So if you go by that kind of a philosophy, definitely A and B are very closely similar because they share 60% similarity. But the cladistics looks at totally from a different way. It doesn't look at the zero states of character. It looks at only the, as I told you, it looks at only the advanced state of characters, where zero state represents the kind of a primitive state, and the advanced state is represented by the 1, numerical digit 1. So if you go by the cladistic principle, you have to take only this into consideration, not these things. So here B and C share the 2 advanced characters, that is character 1 and character 2, 2 advanced characters, and which means they are similar than A. So the entire way that is represented will change based on statistics and based on phonetics. So B and C will emerge as if they are diverging from one ancestor. And here, whereas A and B are similar, there is no kind of a phylogeny that is indicated. You must understand this tree, particularly this tree, does not indicate any phylogeny. It denotes only the similarity between groups. A and B are similar, whereas C is a little different. Whereas this tree indicates B and C have evolved from a common ancestor, whereas A is probably a branch off from another common ancestor, which is down below here. So, and that is how you should be interpreting the trees when you look at the cladistic and phylogenetic trees, especially phonetic trees. Today, of course, there are no phonetic trees that are appearing in publications, only cladistic trees. Right. And the important thing about cladistic is it accepts only monophyletic groups as a strict principle. There is nothing else that is accepted as a group by cladistics. And application of strict monophyly, because that is what we are seeing today, we are experiencing today. Application of strict monophyly has resulted in several changes in classification. Whether it is splitting up of families, whether it is splitting up of genera, everything is due to this application of very strict monophyly. We'll understand as we go into the APG classification. So this 2 things which you must understand. So cladistics accepts only monophyletic groups, and it doesn't accept any other group. Though for sometimes, for convenience sake, it may probably accept a paraphyletic group, but it doesn't accept polyphyletic groups. Okay. To understand what is monophyly and what is paraphyly and what is polyphyly, look at this cladogram. This is, of course, the phylogenetic relationship of different groups of angiosperms. And if I ask you, how do you define an angiosperm or a flowering plant? What are the most important characters of a flowering plant based on which you can differentiate from rest of the plant groups? Because you may remember only a few characters, but remember all these characters are characters that differentiated flowering plants, right? Until here. Okay. So the entire flowering plants, which of course there is no common ancestor that is indicated here, but still let's assume there is a hypothetical ancestor here. The entire group emanates from one ancestor but branches into different clades. So the whole group can be looked at as one monophyletic group. That means a monophyletic group is defined as ancestor plus all its descendants. So if you have an ancestor here, all the descendants of the ancestor form one monophyletic group. And you will have several monophyletic groups in this cladogram. For example, this group from this ancestor, this entire group is a monophyletic group. And this entire group called eudicards is a monophyletic group. And the monocartelidans, that monocards, is one monophyletic group. Look at all monocards are in this single clade. So they don't deviate into any other clades, and all monocards into this single clade. So this is one monophyletic group. And magnolids, all the so-called primitive families, or any other, not so-called primitive families as per our earlier understanding, these are all emerge into one monophyletic group. You can see this, one monophyletic group. Right? So this is what is monophyly means. What is paraphyly then? Here we can see one definite monophyletic group called monocot. This is monocot clade. And all monocots are in this clade. Now, where are dicots? Can you show a single or a kind of monophyletic group that is called as dicots? No. Dicots are distributed in several clades. Dicots are here in the eudicots. Dicots are there in the magnolids. Dicots are there in the ceratophyllales. Dicots are also there in the early angiosperm clade. So dicots are distributed in several of these things. That means dicot as a group is not probably entire things is from one ancestor is grouped into one group. Otherwise, all the descendants are not in one group. They are distributed in several groups. So this is, when you call dicot, you are probably ignoring a few groups and calling a few as dicots. Eudicots or monodicots or magnolids, all these are dicots. So when a situation arises like this is paraphyly, but still we use the word dicot. That's why sometimes we allow or admit some of these paraphyletic groups for the sake of convenience, because dicots are probably largely used in a different way in several other sectors, especially in anatomy, and probably for different ways of understanding. We still use these words, dicots and monocots. Strictly speaking, monophyly does not permit the word of dicot. Either you have to call eudicot or probably magnolid or probably anything else. There is no group called dicot as per strict monophyly and as per the understanding of cladistics. So that is the current kind of understanding today. It's a very simplified. I have, in fact, this morning I have passed on this publication to Dr. Reddy, which of course is, he already said he has circulated to all the participants. You please go through this thing, which is a very beautiful publication that has come from a group of botanists who put things in a very pictorial way. There's also beautiful flower pictures which group many of these things based on the flower structures. So it's a very nice publication for you to go through. If you want to read, of course, in details, you can read. But for beginners, I would appreciate if you go through and probably admire the kind of things that are depicted there. Then later on, you get into the reading. The first thing, the problem is we always probably very kind of people who just like to put our things into the center of the thing, which is sometimes is injurious. When you eat probably the porridge, or what is known in Telugu as sangati, if you put your finger at the center, it'll hurt you because it's hot at the center. But if you slowly go from periphery, you can understand and you can go into the center and handle it very easily. That is what I suggest for you to learn when you learn these things, which are probably for the many beginners. Go from periphery and slowly go into the center. So this APG4 simplified classification is, you can see there is a group called early angiosperms, and where you have a number of families put here. All these families are definitely the primitive families. And these primitive families are today, you see, are totally different from what we have learned as primitive families from the early classification systems, whether it is Bentham and Hooker or whether it is England and Prantl. It is not in the same order, whether it is complex to simple or simple to complex. It is not in the same order. It's all mixed together based on different characters. We'll, of course, come to see what are the characters that are there in this. The early angiosperms that way, undoubtedly today, the amborellales, which we don't have in India, which is basically New Zealand distribution, is the most primitive family among the plant world. And that has been established by several evidences, by molecular, by other kinds of things, by anatomy, by all kinds of things. Amborellales emerge as the most primitive family in Angiosperms. And the nymphiales is the second primitive. And this primitive, there is one clade. Of course, I told you the word clade represents branches. Each one is a clade. The primitive clade is known as anaclade, A-N-A. Anaclade is nothing but Amborellaceae, Nymphaeaceae, and Astrobiolaceae. So this is what is known as anaclade. So you can remember easily now. Anaclade, you can even remember Anna. Anaclade is something that denotes most primitive families or most primitive clade in the flowering plants. Then in between, of course, the typical kind of dicards, the monocards are placed. Monocards, as I told you, is one monophyletic group which is diverged. And the entire dicards are probably split into two groups. The early diverging group, eudicards, and rest of the dicards, that is so-called much advanced dicards, are put into two groups, rosids and asterids, and these are further divided into different families. So this is the basis of classification of APG4, Angiosperm phylogeny group 4. And as you see, this doesn't have a single author. Like we have been looking at whether it is Linnaeus classification or race classification or Engler and Prantl classification or Cronquist. This doesn't have a single author. This APG4 is by a group of people across the world. It is not from one country, it is from across the world. So there are a number of people who have participated, even now participating from different countries. The contribution is probably so very nil only from India. It's very, very minuscule from India. But whereas even countries like Thailand, Bangladesh, and Malaysia, there are a large number of groups who are participating in this classification systems. They're analyzing different groups and providing results. Based on that, the classification is updated on a regular basis. So that's why yesterday I was discussing with one of my students who was studying the Eratostema group. So I had a problem with one of the plant. So I called him and asked, what is this? Is it Elastema? Is it something else? No, sir, he said, it's not Elastema. It should come under some other genus. And I said, you please look into the current websites. They are putting it under Elastema. Then he got back to me and said, you're right, sir. Last week it was under this genus. This week it is under Eratostema. And probably next week it may change into something else. So it is updated every time. And sometimes this updating causes some irritation to many of the botanists. But we cannot take science that way. We have to probably take this updating things and we have to probably follow things. There are different reasons for it. We cannot ignore science behind this. Simply because it is inconvenient to this, we update our mobiles on a regular basis, right? If a new Android app is coming, of course, immediately go for that. Or if anything else, any new mobile is coming, model is coming with new features, we try to change it. We update with gadgets, but we sometimes feel difficult to update with our knowledge. So that is, which is an attitude that is required to look into this part of science. So asterids and rosids are the two major groups of the eudicards. The APG again has a kind of a wrong understanding, particularly in India. Several people ask people who talk about APG, can you identify with APG? Can the molecular taxonomy be useful in identifying species? So these are some of the questions. Without a basic understanding, what is a classification for? It is not a key. APG is not a key. It is a classification. A classification is meant to provide the understanding of different groups and relationships between different groups. That's what the purpose of a classification. Can you identify with the Cronquist classification? Or can you identify with the Engler and Prantl classification? Or can you identify with Kubitsky's classification? If these questions are asked, it'll be difficult for other people to answer. But of course, you can identify with Linnaeus. There's very simple parameters, just 1, 2, 3. That's all. So it is not possible to equate a classification and the identification. A classification will aid identification, and that is why you have keys. Any classification will provide you keys to identify, and you can construct your own keys. And all the keys, and that's why we call keys are artificials. So artificial key to identification of plants. So you can construct keys, and there are keys that are constructed. For example, Light from Hutchinson was constructed a beautiful key to classify all the flowering plants of the world, at least according to his knowledge. And today it is still useful. So such keys can be constructed very easily, and any flora will give you a key. Whether it is based on Hutchinson, whether it is based on Engler and Prantl, or whether it is based on Bentham and Hooker, any flora will give you a key. Today, floras have started coming with APG classification. The Udupi flora was updated completely, and it is in accordance with the latest classification of APG. And the families are keyed. Each genera in the family are keyed, and within the genera, the species are keyed. So it's easy to identify with keys. So don't think classification is going to help you in identification of keys. Classification is only to understand how things are grouped and what is the relationship between the different groups. That is the purpose of classification. We very often probably confuse between classification and classification. So that is somewhere we have to set right our own mindset. And if we understand this basic thing, then we will not have such questions. Sometimes to me appear very stupid. So if you look at this, I told you, there's again another kind of misconcept about APG. The APG classification is molecular. Yes, it is molecular in a way, no doubt. We have to admit it is molecular. But these molecular characters run as a base. They don't run in the forefront, right? Run as a base. What you store in your kitchen will not be visible from your door. Main door is something, and what is inside your home is different. So this is something which is basis. If you look at the characters that define these groups of these clades of APG, early angiosperms, magnolids, monocots, comelids, and eudicots, and other things, look at these characters. For example, let me just read one group. Early angiosperms, which are woody, vessels mostly, of course, woody, vessels absent, eustele, nodes present, sieve tube plastids with the starch grains, leaf simple, persistent, entire, flowers parts free, strobilar, that means they have a cone structure, perfect, and parts varying, often in threes, weakly differentiated, stamens with broad filaments, pollen monosulcate, gynoecium apocarpus, nectar is absent, embryo very small, aromatic terpenoids. Look at how many different kinds of fields play in this. There is anatomy, there is morphology, and of course there is also palynology, there is embryology, there is phytochemistry. Everything is there as characters for identification. A group is recognized based on these states which are in the forefront. Whereas in the background, it is the molecular data. Molecular data will not be visible to you. And what is visible to you is only the characters that you can infer and you can probably study, you can look at, or you can experiment. So this is what is APG. And molecular, of course, is there to define some of these groups. It runs at the background because only based on molecular analysis, you can differentiate groups along with so many characters. So it is not completely molecular based. It is based on almost contributions from every field of biology. And it's a very composite way of looking at and very comprehensive way of looking at. So if there is any misconception that APG is completely molecular, we cannot identify plants, only you will see ATGC, and there is nothing else. It's long list of codes. No, these are not to be looked at that way. And much more utility is there looking through this way. And this is what we need to understand what is APG classification is all about. If you know this, then I don't think you have difficulty in probably understanding the APG. Right? And of course, the publication which I have shared, I may not be going through every kind of group and their characters to discuss. The publication I shared with Dr. Reddy this morning gives you all kinds of groups and all these details. So I advise you to go through the anaclade, which is the primitive clade, plus magnolids and chloranthales. Everything put together is the early angiosperms. Right? If you go to the earliest thing, the anaclade, magnolids, and chloranthales. This entire thing is the early angiosperms. Right? Is it not easy? And what are the characters that define these early angiosperms? These are the characters. I will not run through these characters, which of course many of which you know well. So these are the early angiosperm characters, which are not probably as per our understanding of early angiosperm characters, like more floral parts, free floral parts, thick floral parts, fleshy floral parts, and bee pollinated. All these things are insect pollinated, in other words. All these things are probably absent here. So it's a different kind of looking at, though there are some of them do appear here, like more floral parts and spiraling of floral parts in trees. Some of them appear here, but there are other characters which do not figure in the early way of understanding what are so-called early angiosperms. And also, probably I want you to remind here, the terminology is here. It is not primitive angiosperms. It is early angiosperms. So the word primitive is a social connotation. I mean, when you think primitive, it is something that is least. But that's not the way probably to undermine the plant world or demean the plant world. So this is a more dignified way of saying they are early angiosperms, they are early people, early ancestors, right? So, and some of these things are all probably also the social sciences playing a role into this in defining our terminology. Early angiosperms is the term that is applied today. No more primitive angiosperms. So this is the group of early angiosperms. Amborellales is undoubtedly the basal clade. The basal clade, Amborellaceae, is the family, and that is the only basal family. If you look at the bottom of the evolution of angiosperms, it begins from Amborellaceae. That is the bottom. And the nymphaeals, which of course has two families earlier, of course three families, the Cabombaceae, Nymphaeaceae, and Hydrothelaceae. So far we never had, of course we know of course these two families, Cabombaceae and Nymphaeaceae, that are represented in Indian flora. Cabombaceae, you will find it often in many places, sometimes cultivated in the aquariums and also as a aquatic plant. Nymphaeaceae, which you know very commonly there in India. So many of them are introduced, only very few are native. But these two families are well known to us. This Hydrothelaceae was totally not known to us from India until one plant was Trithuria that was discovered from Konkan region by Dr. Yadav and Dr. Janardhanam. So this plant is a representative from India, and so far this family was known only from Australia and New Zealand region. So it was not only a new discovery for India and also something which is a very important discovery in terms of phytogeographic distribution. So this is something which we need to understand. Nymphaeaceae, Cabombaceae, and nymphaeals. Astrobilaceae, that anaclade I told you, anaclade. So Astrobilaceae, Trimeniaceae, and Schizandraceae, these are the things. So there are a number of other things like now Piperaceae. If you remember the structure. This is very important for us to understand what is primitive and what is not primitive, and what is the so-called Bentham-Hooker system of primitive concept and Englerin system of primitive concept, plantal system. And here, both the concepts are mixed in terms of the early angiosperm concept. You find the Amentiferae of Anglian plantal coming in the early angiosperms, and you also find some of the Laurels coming into flora. All these are Anglian plantal. And if you look at some of the early Renales or early Benthamanoukal primitive families coming into this, so it's a kind of mixed group based on a different set of characters. Otherwise, what circumscribes early angiosperms today is a totally different set of characters. It is not the characters which we have learned in a traditional way. So we need to reorient our mind to look at the early angiosperms from a different set of circumscription. Monocots. Monocots, I told you, is one monophyletic group. And see, this is the ancestor here. It's one monophyletic group. And this one monophyletic group, but even within monocots, Camelinids is probably as a kind of a separate clade, though it is probably nested within the group of monocots, but it slightly deviates from the rest of the monocots. And similarly, of course, this is a Camelinid group. Camelinid group has three groups: the Camelinales, gingiberales, and poales. All these things are into Camelinids. Early diverging eudicots. That means some of the so-called primitive dicots, which of course we have been studying, and those who have been studying the phylogeny or probably the evolution of angiosperms, you must have been studying ceratophyllales as a kind of linking group between monocot and dicot. I think nowadays, of course, I don't know whether your syllabi has some of these things, but I would advise many of the students to go beyond syllabi and start reading some of these things. So ceratophyllales, and many analyses also show ceratophyllales as one of the immediate plant group for land plants. So, and also for angiosperms. So that is ceratophyllales coming as a group here, a different clade. And Ranunculaceae, which of course is one of the most primitive, or at least a primitive kind of a group in Benthamanoukal. Now it's slightly put away from this early angiosperms, which is equivalent to primitive angiosperms, and appearing in a different place. The same thing with the rest of the groups. Now, I will not be going into the other groups like what we went through, the early angiosperms, monocots, and early diverging dicots. Of course, we will go through what this APG has done in terms of classification, how it has probably changed the way we looked at certain families, the way we understood certain families, and of course, the way we practice the classification. And one of the change that has happened is in the family of Malvaceae. Malvaceae, of course, traditionally has been studied as a separate family, separate from its allied families, the so-called Sterculiaceae, Teleaceae, Bombycaceae. And we have characters, because if you probably, especially the senior faculty members might very well remember, these families were taught as separate families with the characters distinguish all these families, and then there were keys made, and sometimes confusing specimens were given to them in practical classes to confuse them so that they will fail in their identification. All these things were there in early systems of examinations. But today, things were made easy for you. Whether they have given Bombax, or whether they have given Gruvia, or whether they have given Sterculia, you just write the family Malvaceae, you are right, you should be given marks. So is it not easy for students? So it is given a kind of a role to bring several things together into one family. Why this has happened? We have to understand the basis. If you look into this clade, this is one monophyletic clade. You can easily understand why we say this is one monophyletic clade. Everything begins from here. So there's another sub-monophyletic clade here, and this is one major monophyletic clade here. Tell me, what is Malvaceae here? Is Gruviidae Malvaceae? Are Sterculiidae Malvaceae? Are Teleidea is a Malvaceae? No. Everything falls into one monophyletic clade. In the beginning, I told you, anything is defined based on strict application of monophyly. When everything is one monophyletic group, how can you identify Teleaceae here and Sterculiaceae here and here and Malvaceae here and Bombycaceae here? It's very difficult. So that means you are trying to practice paraphily here. When you say Malvaceae, you include only this. You're not including probably other groups. So you include only part of the descendant of an ancestor, which strictly denotes paraphily. Whereas when you include all the descendants of an ancestor, which is monophily. So as per the cladistics, it's the monophily that should be adhered to. So accordingly, it is one Malvaceae. There is no Teleaceae, there is no Sterculiaceae, there is no Bombycaceae, and it's all Malvaceae today. So it's Malvaceae. Sometimes if you want to understand, for your understanding, if you want, you put Malvaceae sensu lato, which means in a broader sense, right? So that is, some people practice. There is no harm in it. So, and that is what is Malvaceae today. And that's what APG has done to the classification. And Fabaceae, we have been classifying Fabaceae, I mean, in a larger sense again. The term Fabaceae could mean in taxonomy only Papilinidae or probably the entire Leguminosae, depending on how you employ. So the Fabaceae or Leguminosae, I would say. We've been looking at this family only from 3 different subfamilies. Subfamily Papilinidae, Cisalpinidae, and Mimosidae. That's our traditional way of looking at. Even I have taught students like this, but today it's something which is totally different. So it is now divided into 6 subfamilies. So these are the 6 subfamilies, and many of them are probably it will be new to you, but this is how today's Fabaceae is divided. And the Fabaceae, of course, I said in Tamil Nadu, but probably in Andhra also probably these are the 4 families, 4 subfamilies that are represented by in this family. Other families we do not have. So the genus Bauhinia, which we have been traditionally studying as under the Cisalpinidae, now goes under Cercididae. And the Ditariidae has some of these genera like Saraca, Tamarindus, Hardwickia, and other things. So Cisalpinidae, the Mimosidae, is being reduced to a small subgroup under Cisalpinidae. There is no separate big Mimosidae. Several genera have undergone changes as per monophily. And if you look at the genus Dysmodium, the Dysmodium is something which is greatly undergone a change into different genera. Today there is Dysmodium is no more Dysmodium. It is split into several genera. Like one species goes into Alicicarpus, some of the species goes into the Buffodia, and a few go into the Monathrocarpus, and at least a group of species grow into Grona. So, and a few species grow into Sohimea. So this is something which is happening to several genera. The same way Cisalpinia. Cisalpinia also, it is either Glandina or Libidibia or Mesoneura or Biancia. For example, Cisalpinia decapetala and Cisalpinia, they go into Biancia, and Cisalpinia mimosidis go into Haltholia. So this is the way that the genera are split today based on strict monophily in APG. The family Apocynaceae and Asclepiadaceae again are traditionally treated as 2 different families. Still, in many parts of India, they study as Apocynaceae and Asclepiadaceae. Still, many floras, of course, still are which are written and which are being written, follow Apocynaceae as a separate family and Asclepiadaceae as a separate family. But this is in much deviation with what the globe follows, what the world follows. And today what the world follows is, see, look at this entire thing is one monophyletic group, which is Apocynaceae clade. So it is no Asclepiadaceae today, only Apocynaceae. So your Calotropis is within Apocynaceae. It is not Asclepiadaceae. If somebody writes Calotropis or Pergolaria as Apocynaceae, they are quite correct according to the current way of understanding. They are not wrong. A major change has happened in the Lamiaceae Verbinaceae circumscription. So this major change is something that is probably for many people is not digested properly. And some people say, how can it be? How can Tectona can be called as Lamiaceae? It's a huge tree, whereas Lamiaceae are small herbs. So this kind of traditional way of characterization or probably circumscribing the families are still stuck in our mind. We have to probably de-learn some of these circumscriptions and relearn the new circumscriptions. And this is the Lamiaceae Verbinaceae clade. So if you look into, there is no one monophyletic Lamiaceae clade, and there is no one monophyly the Verbinaceae clade. Everything is split into. So, and that is why the whole thing is one monophyletic Lamiaceae Verbinaceae clade. So how do you differentiate Lamiaceae and Verbinaceae? This is the new circumscription for Lamiaceae and Verbinaceae. Earlier ideas about like trees and herbs and probably the other characters, so now they are no more in use. One of the best and easy character to distinguish the Lamiaceae and Verbinaceae as per the present concept is this, the inflorescence character. If the inflorescence is verticell, thyrsus, cyme and panicle, it is Lamiaceae. If the inflorescence is racemose, just like you find in Stachytarpita, it is racemose, it is Verbinaceae. Tell me now, the Tectona, whether it has a racemose inflorescence or panicle inflorescence. So it is panicle. It cannot go under Verbinaceae. So, and the same way, many plants which were under Verbinaceae are brought into Lamiaceae based on this major circumscription of the family that defines what is Lamiaceae currently and what is Verbinaceae currently. The corolla, it's conspicuously bilabiate, whereas in Verbinaceae it's weakly bilabiate. Exine, not thickened near aperture, whereas it is thickened near aperture. And style, it's clearly bifid, not very clearly bifid. Stigma, of course, it has stigmatite tissue, inconspicuous and very conspicuous stigmatite tissue. So those who have seen, of course, Stachytarpita will remember it doesn't have conspicuously bilabiate corolla, but those who have seen other plants like Vitex, it has very conspicuously bilabiate corolla, so it should come under this. All these genera which were originally treated or conventionally treated under Verbinaceae are today under Lamiaceae as per APG. So whether it is Premna or Symphorema or Tectona or Vitex, everything is under Lamiaceae today. So don't get probably frightened when somebody says Melina is a Lamiaceae member, a huge tree. Yes, it is a Lamiaceae member because of the characters which I told you in the beginning. Some of the families, of course, have undergone great changes beyond our probably understanding or beyond our kind of comprehension. Liliaceae is one such family. Today, there is no Liliaceae. In fact, we had encountered a kind of a problem when we were engaged in writing a the +1 and +2 textbooks for Tamil Nadu. Teacher said, sir, Liliaceae is included in the family. Then what is the example that you're going to give in the Liliaceae? They said it's usually allium, which is onion. Then I said they cannot go together. Whether you want Liliaceae in the syllabus or whether you want allium to come in the syllabus. If you want Liliaceae in the syllabus, allium cannot be in the picture. If you want allium as an example, it cannot be under Liliaceae. So now today's Liliaceae is split into 14 families. The things like genera like Asparagus, Agave, and Scilla are all under Asparagaceae. Gloriosa, Colchicum, and Iphigenia are all under Colchicaceae. Allium, Crinum, and Agapanthus, they're all under Amaryllidaceae. Allium goes under Amaryllidaceae. And what is Liliaceae sensu stricto today? That means in strict sense today, are 3 genera: Lilium, Fritillaria, and Tulipa. Among these 3 genera, Fritillaria and Tulipa are genera which are alien to us. Though we are sometimes it may be cultivated. Tulips, for example, are cultivated in Kashmir, but we don't have natural flora here. Whereas Lilium, we do have a few species which are native to India. Particularly in South India, we have one Lilium niggerans, which is quite endemic. So when we say Liliaceae, we are talking about only these 3, whereas other genera are under different families. So we should be able to grasp some of these things. And then, if you go through this Colchicaceae, Gloriosa, Colchicum, and Iphigenia, all of them are characterized by a presence of chemicals, certain kinds of chemicals like colchicine and other things, which you know the use of them, which are not only used in cell biology, also used in the pharmacology. The generic concepts are changing, not only family concepts, the generic concepts are changing. And Polyalthia, we know, of course, traditionally as a genus. And we have been, of course, we know several Polyalthia species. One of the well-known species is Polyalthia longifolia. All of them are very popular to Polyalthia longifolia, which are commonly cultivated throughout India. Any part of Andhra Pradesh you go, you will see this plant. Or any part of India you go, you see this plant. But today, Polyalthia is not a monophyletic group. It is split into several polyphyletic groups. So all Polyalthia species, so it has to be split into many genera. That is when these things come. That's good. The Polyalthia today is split into all these genera: Phenirevia, Shivarantha, Massia, Marsipopetalum, Munoon, and Polyalthia sensu stricto. See, for example, your Polyalthia cerassoides falls within this Polyalthia. I mean, what we know traditionally as Polyalthia, it includes Polyalthia cerassoides. Whereas Polyalthia longifolia is now Munoon longifolium. So it is no more Polyalthia longifolia. And Polyalthia corinthi, now today Huberantha corinthii. So the Polyalthia species have gone into different groups, and these are the split genera under which they go and nest. So it is not a monophyletic group, and whereas these genera are monophyletic groups as per the cladistic analysis. So we should probably recognize these monophyletic groups as per the principle of cladistics, and when we apply these groups, so we need to go for these changes which are inevitable. And the same way, Cleome. The genus Cleome is a different genus today, which does not include our Cleome viscosa, which we commonly call as Cleome viscosa, and several genera of Cleome also. For example, the Aspera, Farina, and Cheridonae, they also go into different genus. Look at this. You have this a cladogram, and look at this. There is a Cleome sensu stricto clade here, this one monophyletic group. And there is a Cleomella clade here. This is one monophyletic group. Right? So like this, there is a Corinandra clade here. This is one monophyletic group. So like this, this is an Arivela kind of a clade, and this entire clade is Arivela clade, which is one monophyletic group. So if you apply these monophyletic groups and try to understand the genera, the Cleome viscosa should go under Arivela, Cleome aspera, Felina, and Cheridonae should go under Corinandra, as per Indian Cleomes are concerned. And similarly, genus Molugo. Molugo is not one monophyletic group, and it is probably split into several monophyletic groups. So accordingly, this Molugo sensu stricto consists only Molugo diastica. Genus Paramolugo, which has Paramolugo nudicalis, that is former Molugo nudicalis. Genus Trigastrotheca consists of Pentaphylla and stricta, that is former Molugo pentaphylla and stricta. Genus Hypertellus has Molugo cerviana. Now, one of my students, of course, has further analyzed and created one more genus, which is to be published very soon, called Paleomolugo. And it has one or two species into this. So it is constantly getting updated. So it is not something that is static. Whereas the kind of things which we have studied, they are static for a long centuries, long years. But now things are getting updated, getting changed every day and every week, and things are appearing in the net. The same way Hybanthus today is Afro-Hybanthus. Hybanthus Angiosperms is now forms a species under Afro-Hybanthus, and it is split into few genera like Pombalia, Cubelium, and Pygia. The Serapygia, the Carolumas and Brachystelmas. Particularly Brachystelma, there is a huge diversity in Andhra Pradesh, generally in dry regions. Same thing with Carolumas. But now these two are not distinct genera. They are no more distinct genera. They've been based on the molecular analysis, based on cladistic analysis, have been clubbed into one genus by Bruins from South Africa, who just, of course, very recently, just a few weeks back, he passed away. Serapygia now consists of both Caroluma and Brachystelma. So there is no more Caroluma, no more Brachystelma, all under Serapygia. These changes have to be reflected within our systems. And of course, several floras are now following these changes. If you look at our history, the history is something from here. Although I did not go earlier in the history, like right from Theophrastus, in terms of families, we begin here. Bentham and Hooker, we started our learning of plant classification with 197 families, then moved to England and Prandtl with 303, Cronquist 389, APG 462, and APG 2 457, APG 3 413, and 416 families in APG 4, but we don't know where we will move again. So the learning curve should be from here to here, whereas we are yet to move from here. In most of the countries, it has made a jump. They were all here, of course. Even Europe was here for a long time, using only England and Prandtl. And once the APG came into being, they jumped from here to here, and maybe trying to move further. Whereas we are stuck here. We are unwilling to move from here. And saying, of course, giving several reasons which are not acceptable reasons. Like what I said, can we identify, or can molecular basis identify species? All kinds of excuses we are giving, because we are not, or we do not want to learn new things, or we do not want to probably enter into new arena. That's the problem. So, and that is the reason why we are still stuck here, whereas the whole world has moved here. So that's, otherwise I would say we are still primitive in our approach. We need to move from this place to this place in the ladder and probably move further. Every day it is getting updated, and every day the families are changed. Every day the new families, of course, either getting merged or probably certain families are being created, if not every day, at least periodically, I would say. Every day is just one kind of a phrase that I use to denote the periodicity. So changes are happening. Why not we move with the change? When we move with the change in rest of the things in our life, why not we move with the change in this system also, in the classification also? And that's my request. We need to move ahead. We need to move with the times, and sometimes we need to move ahead of times. And as I told you earlier, there is very little contribution as far as the APG system is concerned from India. And there are very few biologists or botanists who are probably trying to work. Of course, today the present generation is coming into being. I told you one of my students creating a new genus. So a large number of youngsters are today trying to come into this field of cladistics, trying to use and trying to study several groups using cladistics. So that is a major kind of change that I see that is happening among youngsters. Whereas the universities and colleges are yet to change in a way, in the sense, though researchers are working on it, the syllabi has not changed. The syllabus has to change from what has been traditional, artificial, natural, and phylogenetic. It has to change from phonetic to phylogenetic. All the phonetic systems, let them be learned as part of history. Whereas the phylogenetic system, the APG3 or APG4 better, another version might come very soon. So APG4, let it be learned as the classification system in the botany, for the plants. Let the youngsters probably be thrown into this. Let the youngsters be introduced about these systems. And that is what probably will make our science strong. Otherwise, sitting here, we will be probably mocked by the rest of the world. What are we saying? In fact, this has happened in one of the Angiosperm taxonomy seminars. One of the person who came from Sweden to attend the seminar, when our students are presenting about their analysis of flora, of course, many people study a flora of a region and try to present there in the seminars. When one of the students is trying to present, of course, typically we classify flora as this many dicards, this many monocards, and these many families in dicards, these many families in monocards, the dominant families. And this is a very typical way of doing it in India. So, and when he was presenting that way, the person from Sweden stood up and asked, why are you trying to present your analysis in groups that are not known? Very simple question. So that is how the world is looking at us. Whereas we are trying to give excuses for probably changing from this to this will not help us. We need to change. And in that way, I probably appreciate youngsters who are trying to change, who are doing, of course, the cladistic analysis, who are into molecular taxonomy, either alone or probably in collaboration with scientists from here or from abroad. There are a large number of groups which are collaborating with abroad and trying to publish papers. There are now, of course, papers are publishing. In fact, with Bruins, there's a group from Maharashtra, Kolhapur, who have collaborated immensely and published papers. So, and things are happening that way. I think we need to go from this. We need to take this as examples and move towards the advancement of science, not in a retrogressive way. Whereas we are remaining here in a retrogressive way, we need to progress. That's my request. Thank you.
Yeah, thank you so much, sir. It's a wonderful session and a terrific lectures. Continuously 2 hours lecture. Very informative session, sir.
Thank
And some of our participants are eagerly waiting to interact with you, sir.
Sure.
Over to Sadhana.
Yes, sir. Thank you, sir. Everyone who wants to interact with sir or ask questions can please raise your hands. Whoever wants to pose questions, please raise your hands.
Muhammad Mustafa.
Hello, sir. Good afternoon. It's a very excellent talk given by Dr. Narasimhan, sir.
Thank you.
So, this is Dr. Muhammad Mustafa from Kakati University, Associate Professor. Sir, you have mentioned clearly about Taktajan's classification.
Yes.
Actually, Taktajan, he was the very authoritative on phytogeography and evolution.
Yes.
However, he followed phonetic system.
Yeah.
His classification is a wonderful classification.
Yes.
It's a milestone in the history of phylogenetic system of classification.
Sure.
Actually, he first time when he published his classification in the year 1989, he mentioned only 4 subclasses under Liliopsida and 7 subclasses under Magnoliopsida.
Yeah.
Later, when he revised in the year 1999, he has mentioned clearly about under Liliopsida 11 classes and under Magnoliopsida 11 classes, under Liliopsida 6 classes. Totally 17 classes were there, sir. But in our slide, it is mentioned that is the primitive one. That is the first time when he mentioned about this classification under his book, that is Diversity and Classification of Flowering Plants.
Yes. So I agree with you. There is no dispute about that.
Yeah.
What I try to present here as past history in a schematic way. Because details about each of these classification we cannot go through in a large perspective manner, because otherwise I won't be able to cover the entire history.
Yes.
So that is why I present it in a schematic way. So where I have given only skeletal details.
Yes.
And these are the major things, of course, which he put forward in the initial time. Later on, of course, even Grandquist also has expanded it later. So everybody, Kubitsky initially followed the Taktajan style. Today he follows the APG4. All his publications, books that are coming up, is based on APG4. So yes. So that is why I tried to provide you the gist of the classification in a schematic way, not expanding the classification, so that it takes time for me and more slides, so that I may consume more time. Even this, as Redigaru puts it, took 2 hours.
Yeah. Under APG classification, both the Asclepidaceae is totally merged into Apocynaceae. That work was already done by him.
Yeah.
Based on the Z and at all.
There are examples, sir. There are examples like this earlier also, where many families, like already there was a concept of larger Malvaceae that was coming up, and there were examples of a few families that were merging, and there are examples of few genera getting split. But these things have happened only based on without molecular basis. Now, molecular basis provides a very clear-cut way why we should probably divide these things and consider as monophyletic groups. This monophily is missing in earlier kind of merging and dividing.
Hydatillaceae alone representation examples.
No, Hydatillaceae. Yes, Hydatillaceae is one of the primitive thing.
Primitive one. Example of India, one and only one.
That is Trithuria from India. Trithuria, and Hydatella is from New Zealand.
Thank you, sir.
Yeah.
Over to Dr. B. Jyoti Garu. You can pose your question to the speaker, madam. Somebody like Realme 5.
Hello.
Yes, Jyoti ma'am, carry on with your question, ma'am.
Good morning to everybody, sir.
Morning.
Sir.
Yeah.
Sir, under undergraduation level and PG level, which classification have to be followed, sir?
This is a big question that most universities have. Right? So I understand the kind of dilemma that many teachers have, especially in putting in the syllabus. As a
Yes,
teacher,
sir.
what I did was,
Mm.
in my college, when
Yes.
I was trying to change the syllabus, initially, of course, 10 years ago, I had Benjamin Hooker for the UGs and later for PGs, APG. But now I changed even the UG into APG. Not only that, recently we have also used the APG system in the +2 textbook. We have brought this into +2 textbook at a primitive level of introduction, at an introductory level. So things have to move forward. We cannot be, because what the problem is, when you discuss the old systems again and again, they get probably re-emphasized, they get probably re-stressed, and people will not move from those things to new things. So we need to change. The quicker the change, it is the better. And don't bother about whether people will learn or not. People will learn. Now, +2 students in Tamil Nadu are learning the system. You can download the textbook from the Tamil Nadu Textbook Society and look at the way in which it is done. So it is something which is very important to introduce these things. And elsewhere, it is learned much earlier. And most of the universities abroad, they don't have Benjamin Hooker or Engler and Prandtl systems in the textbook at all. It is only one system, that is the present system, classification system. So we need to probably, when we emulate many of those things from abroad, why not do this? So introduce the system to the UG itself, I would advise. But teach them in a probably skeletal way. Don't teach them in a detailed way, but let them understand what is the system. Let them understand what principles it operates. Then when they go into PG, probably they can study it in more detail. That's what I would suggest. But the university should be bold enough to do this change. There will be several oppositions from staff members which will be coming. How will our university UG students will understand they did not study this? All these things will be coming. And they are only excuses from staff members, not from students.
Dr. Paramesha,
So I'm sorry for this blunt statement, but that is what it is.
Dr. Paramesha Mallegowda also had the same question, so I think his doubt is also cleared. And next, who else
We have
wants
somebody
to ask?
named Unread Blue, right? So they can ask the question.
Sir, good morning to everyone, sir.
Yeah.
Sir, what is the major difference between Asclepiades and Apocynaceae?
Now there is no Asclepiades and Apocynaceae, madam. It is all one. So I don't want to dwell into differences, but there is it's all one monophyletic clade. That's what I would say.
Okay, sir.
Typically, of course, you would have learned differences, right? How styles merge, how styles do not merge, all the other kinds of things, right? And presence of the elaters and all kinds of things. But now there is no difference because there are intermediate things and there are other characters that unite them. So now it is one group. It is only Apocynaceae, no Asclepiades at all.
Sir, which classification we have to follow, sir?
I told you, please follow APG. That's the only thing that can put you on par with the world.
For intermediate.
Even from intermediate, that's what I'm saying. Even from intermediate, please follow.
Okay, sir. Thank you, sir.
Yeah.
Sir, Bhavani Gonuguntla is asking, APG classification is still undergoing transformation. So will there be any end to it?
See, that's what it's a dynamic system. And taxonomy is something that is dynamic. It cannot be static. And the history that we went through also tells you how it is dynamic in each period, during each period, right? So each period here, of course, certain periods lasted for a few centuries, certain periods probably last for only months. Now, when you consider the current period, because the advancement is so much and several people are working from several quarters of the world and the information is pouring in. So it happens, but we are almost nearing towards some kind of agreement with most of the changes that have been made. That's why not many changes are appearing now. So otherwise, if the changes are quite fast, another version would have appeared very easily by now. But the next version will take time. So now we are almost stabilizing.
Sir, good afternoon, sir. This is Dr. Akbar, lecturer in botany, sir, from Andhra Pradesh. Could I speak to presenter, sir?
Please.
Hello, sir.
Yeah.
Yeah. Is it possible to incorporate practicals, or else how can we conduct the practicals if APG system classification is followed? Say, for example, how can we explain the students our general families like Anonaceae, Magnoliaceae, Liliaceae, like that, sir? Is it possible to conduct the practical, sir, for the students of graduation at least?
There is no harm in conducting the practicals using APG.
How can we conduct, sir?
No, let me tell you, sir. Let me tell you,
yes, sir.
Mr. Akbar.
Yes, sir.
See, that is another myth that we have. I would call some of these as myth, are blocks in our mind. I'm sorry to use these words, but still, see, when you conduct practicals, you give a specimen, maybe from one family, let's say Malvaceae or Anonaceae, right? If you suppose, imagine you give a family, maybe you give a Gruvia specimen and write it on the board, the details, Gruvia hirsuta, family Malvaceae. What's wrong in it? The student is going to dissect the specimen, understand the characters, and already the characters for larger Malvaceae available in several floras, go by that. And our problem is also the textbooks that we follow and the reference books that we use. We are not trying to use the latest textbooks. We do not have enough stock in our libraries. For example, I don't know whether your college has the plant dictionary by Maberly, right? Which is the updated dictionary, which has all the updated concepts. And Malvaceae is defined based on the current phylogeny concept. So, and it lists characters. So why don't we redefine our families as per APG? Then what's the harm in conducting practicals as per APG? So it's not going to be difficult, sir. It is our mindset. We need to change.
Any more questions, participants?
Somebody is in the panel. Android Blue Ride, you had a question. You can please unmute your mic and pose the question to the speaker.
It's a nice name, Android Blue Ride. Very peculiar name. And look at how names are getting updated using technology.
Okay.
Right, this is one example. This is one example where tells us how we should update ourselves.
Sure. Madam Megashri Garu, you can ask your question.
Thank you, sir. Sir, I am from Mysore University. I am a student.
Good.
And
Good.
Also now I am a research scholar. The problem is getting the information about APG system is very difficult because it is not available in any common textbooks. How we get this information or updates about this APG system?
Very good. That means the common textbooks are at least a decade old in India. Right. I mean, decade is simply one period. Probably it is much older than a decade. Because what many of the run-of-the-mill textbooks that we have in India are all written by people who are probably not taxonomists by themselves. So that is the problem. But we have two good textbooks which are based on APG, written by one by Indian, which is Guru Charan Singh. And that book is available now in many bookstores. I don't know whether your university has. Guru Charan Singh has two versions. One is a abridged version. The other one is a larger version. Larger version I would suggest because it gives much details. But
sir, one thing is I am following that Guru Charan Singh textbook for
Guru Charan Singh, smaller version probably, abridged version.
Yes,
not
sir.
the larger
And Systematics by Guru Charan Singh. I am following the textbook only.
Yeah.
But
Okay.
for getting more information or updates, which website will provide the information about APG system?
MOBOT, that is, you go for MOBOT, that is molecular phylogeny of biology. I'll send you the website probably very clearly to Dr. Reddy Garu. We can pass it on to the participants, the link. So that gives you every update that is happening and all the details about families and other kinds of things happening in APG. But also there are other textbooks apart from Guru Charan Singh, which you may follow probably a smaller version of Guru Charan Singh. The best textbook I would recommend always to many colleges and many universities is Plant Systematics by M.G. Simpson. I do not know whether your colleges have this textbook, which is costlier, probably costs about 5,000, 6,000 rupees. But still, it is worthwhile to get this book. Right?
Okay, sir. Thank you so much.
In fact, I have advised many teachers to buy individual copies and people have bought copies.
Thank you, sir.
Welcome.
Sayed Siraj, please post your question. Unmute your mic and please post your question.
No, he has been continuously raising his hand. I think he might not have a question. Sir, we are done with the question session, sir. Parmesh Lingala is there. We'll allow him. I think this will be the last question.
So good afternoon, sir.
Yeah.
Sir, I have a small doubt. In internet, there are different types of browsers are there. Yes. Sir, in internet, I am searching for the author citations and update of names. The plant list and POVO, plant suffered online, and IPNI and catalog FLI. All these are I am following. Yeah. But which one I need to follow, sir? At which one is now recently modifying and changing their names according to APG, sir? Which site is better for this?
Yes. But I would say you should be very cautious about in following any of these things. Don't blindly follow. Even plans of the world online also has a number of mistakes.
Yeah,
right?
sir. Yeah, that is the main doubt, sir. I have ambiguity to follow which one.
I know, number of mistakes. And the plant list is now getting, of course, updated. For some time, it was not updated. For nearly 4 or 5 years, it was not updated. So you have.
This is the problem for the young botanist.
Yes, for that, you should understand the plant and the taxonomy and the nomenclature. If you are very clear about it, then you can choose the decision based on what the websites provide, right? I'm continuously referring for the past 4 months. We are now preparing the list of plants in Tamil Nadu. So we've been referring to these websites. I find large number of mistakes also in the POVO and other kinds of things. Whereas IPNI provides only the author citations and publication details. It does not give you what is right, what is wrong, what is probably correct name, what is synonym, all these things. It never gives you kind of decisions. But whereas other things will give you decisions. So you need to be very careful in following these decisions in accordance with your taxonomy knowledge, in accordance with what generally, and also consult experts. If you're not able to fix, consult experts. What is the kind of things? So that's what we do.
Sir, we are done with the questions, sir. Over to Vijayabhaskar Reddy, sir.
It's a pleasure to propose a vote of thanks to Dr. Narasimhan, sir, on behalf of organizing committee and present attendees, and also on my own behalf. Really, sir, it's a wonderful session for us. You elaborated in such a way from basics to applied and history to recent. And many of change is inevitable. What you said is 100% correct, sir. Many of our faculty members are in a junction till today. They're looking to choose which system has to be. And you're the person given a clear-cut idea, I think. Now, many of participants got the idea to choose because change is inevitable. We have to move ahead. And according to the change, we should adapt to the system and move ahead. Otherwise, we will perish. That's a motto we received from you, sir. And we also thank for your sparing very valuable time and high knowledge with our participants. Really, we motivated today, and many of our participants inspired and posting their compliments. Really, sir, we enjoyed a lot and we learned a lot. In future also, we need persons like you, sir. Please, whenever we request you, please accept, and further more sessions we require from your side. And morning also, you shared the document, very valuable document. Immediately we shared with all participants. We got a good response from them. And possible, many are requesting your PPT presentation also. Possible, you share us, then definitely we'll share to our participants, sir.
Sure.
And once again,
by mail.
Yeah, surely, sir. Thank you so much. Thank you. And because of your major support we are running all the sessions successfully, sir. You're also a part of this short-term course, actually, VP.
No, it's our duty to probably inform people and spread information, whatever that we have learned.
Yeah.
And there is similar-minded people are there throughout India who like to share.
Yes. Because it should continue. The knowledge should be continued and shared. We want to promote the taxonomy. That's the main motto. With that motto, we came out and we started this. Really, sir, with the backbone support.
Not promote taxonomy, promote quality taxonomy.
Yeah,
that is what we are trying to attempt.
Yes, sir.
Quality taxonomy.
Really, sir. Once again, thank you, Nodal.
Thank you, sir. I think from my side, I'd like to thank you very much for calling me and giving me an opportunity to share my ideas. And also thanks for Shashikala ma'am and Sadhana and also Srujan for teaching out some of these things.
Yes, sir.
Thank you all, the entire team. Yes, ma'am.
And ma'am would like to share.
I want to share something, sir.
Yes,
sir, thank you very, very much, sir, for accepting our invitation and all. Anyhow, this is outstanding, sir. But apart from this, what I want to say is we have to learn a lot from you, sir. Because from exactly 10 o'clock onwards till now, without break, you have given very good exhaustive lecture and very informative lecture. So all we, the professionals or the students, we have to learn this thing from you, sir. It is very encouraging for us, to our scholars and also our students and faculty members, sir. These two technical team, Srujan, Sadhana, they are my scholars, sir. So they should also learn this technique, how to give such continuous lecture like this thing. So we are very, very happy to have you here, sir, to encourage our participants as well as our complete team, sir. Thank you very much, sir.
Sometimes continuous lectures will be very boring.
No, sir, it is not at all boring, sir. It was very informative.
All of you would have also probably well experienced in giving.
Yeah.
It's not something which is quality. Thank you so much for your opportunity.
Yes, sir.
Over to Srujan.
Sir, I have a comment for the participants. Thanks for the session. We have posted 2 links in the chat box and in our WhatsApp groups too. The one link is regarding the feedback and the evaluation form. The other link is regarding the contacts. So we want to have a community of plant taxonomists. So if you want to associate, you can write, you can fill in the form, and no column is compulsory. Whichever is good for you, whichever you are, feel privacy, you can share those information. Otherwise, you can skip the form and skip the columns too. No, it is not mandatory to fill all the sections in the contact form, but it is mandatory to fill all the columns in the feedback and attendance form. So kindly make sure that you submit the things. If there is any problem, kindly chat on the WhatsApp. Thank you. And the next session will start sharply by 2 p.m. Okay, thank you.
Thank you, sir. Thank you all. See you. Bye. So shall I leave?
Yes, sir.
Yes, sir. We are going to end the session, sir. Thank you, sir. Thank you, Narasimha, sir.
Welcome.
Thank you all.
Welcome. Right.
Sir, thank you, sir.